Across numerous forms of life, cyclic adenosine 3,5 monophosphate, otherwise known as cyclic AMP, is used in countless cellular processes that include but are not limited to lipid metabolism, sensory processing such as olfaction, and even cell growth and differentiation. If you've taken an introductory biology course or biochemistry, then you've likely had to study this molecule or a pathway it's involved in intensely. But you may be wondering, if cyclic AMP is such an important molecule that's used for so many different purposes, then there must be an equally, if not even more important protein that regulates its synthesis, right? And well, if you were thinking of adenylyl cyclase, you'd be spot on. Adenylyl cyclase is not one of, but actually the sole enzyme that actually synthesizes the conversion of ATP to cyclic AMP. Let's take a look at some of its interesting structural features. Here, through the power of JMOL, we can actually see the structure of an adenylyl cyclase. The protein databank ID for this particular molecule complex is 6R3Q, and it was deposited by Dr. Korkov and Dr. Chi, who were funded by the Swiss National Science Foundation. There are 6 distinct classes of adenylyl cyclases found in the many kingdoms of life, with 10 isoforms in the mammalian class 3 alone. Be that as it may, all 6 classes serve the same function in catalyzing ATP into cyclic AMP, and many classes contain similar structures to properly fulfill this function, as shown by this excerpt from Khan-Panavar et al. But this complex shows the subtype adenylyl cyclase 9, which is a part of class 3, an integral membrane protein used to transform extracellular signals, or cues, into cascade responses within the cell. They're also found in numerous tissues, such as, but not limited to, the lungs, the heart, and even the brain. Shown here in blue is the adenylyl cyclase, and bound to it is a G protein, shown in green. Now from this recolored snapshot, we can observe some of the common characteristics found in all adenylyl cyclases. Firstly, we see the yellow labeled antiparallel beta sheets, which are hydrophobic, meaning they are nonpolar and repel water. We also see that they are surrounded by these magenta labeled alpha helices, which are amphipathic, meaning that they contain both hydrophilic and hydrophobic parts. And like all other adenylyl cyclases, there are 2 different monomers, which are combined to form a homodimer that allows adenylyl cyclase to convert ATP to cyclic AMP. Of quick interesting note, many adenylyl cyclases contain structures known as scolin, which have the main purpose of increasing stability and therefore functionality of the adenylyl cyclase. In that sense, they're sometimes classified as regulators of adenylyl cyclase, like a certain molecule that you may have heard of, G proteins, but we'll get to those in a second. You can also take them as supplements, often used as an asthma drug, and comically, as a weight loss supplement, although there haven't been many studies to prove that it's effective in burning fat. Here we can get a better look at the G protein of this molecule. G proteins are used as regulators for adenylyl cyclase. These G proteins can either be inhibitory, labeled GI, or active, labeled GQ or GS. And yes, even these regulator molecules for other regulator molecules are, as you can guess, regulated by other regulator molecules. A G protein is made active when an extracellular ligand, such as a hormone or neurotransmitter, binds to the G protein coupled receptor. This causes a conformational change in the structure of the G protein coupled to the receptor, where a GDP is exchanged for GTP in the site shown through JMOL. And at this point, the G protein is activated and ready to bind to adenylyl cyclase. Although, keep in mind, G proteins target other molecules and complexes too. As mentioned prior, the activated adenylyl cyclase goes on to catalyze the conversion of ATP to 3,5-cyclic AMP, but it also releases pyrophosphate as a product. Cyclic AMP then goes on to bind to activate other molecules, which is used for a myriad of functions. And what's one of those important functions, you may ask? Well, only something that is required for all of us to grow and survive: cell growth and differentiation. Where protein kinase A gets activated by synthesized cyclic AMP. From there, the active catalytic subunits go on to activate transcription growth factors, and they go on to promote cell growth and division. However, Sometimes a mutation in this pathway can lead to overactivation of the cell division process, and that, unfortunately, can lead to tumors. Luckily, because of the versatility of adenylyl cyclase and the fact that it's the sole synthesizer of cyclic AMP, that leads one to theorize that if we can find ways to control the levels of expression and activity within cyst and tumor cells, then maybe we can find more effective ways in fighting cancerous diseases. Thankfully, there have been a myriad of researchers who are exploring this very possibility and discovering that there may very well be some truth to this theory, and maybe someday this can be implemented into a variety of novel treatments for cases where cellular processes are running out of control, like cancer. And that's all. I hope you were able to learn a lot more about adenylyl cyclase and understand how interesting yet important of a protein it is for many of the functions in your daily life. And here are some of the sources and papers I've used for this project. If you're interested in learning more about the molecule or more about the research that's going on with the molecule, I encourage you to check out some of these papers and more.